It's a great honor to be here today to introduce our team's latest research, entitled The Gut Microbiome Promotes the Growth Performance of Black Soldier Fly Larvae by Detoxifying Uric Acid. Black soldier fly larvae can convert various organic wastes, including livestock and poultry manure, and have thus garnered widespread attention. However, uric acid constitutes approximately 50% of the nitrogenous compounds in poultry manure and is also a primary nitrogenous waste product in insects themselves. Understanding how BSF larvae process high uric acid substrates is an urgent task. Extensive studies report that the gut microbiome is crucial for BSF larvae in waste conversion, leading us to hypothesize that gut microbiome could contribute to the degradation of uric acid in BSF larvae. First, we confirmed that BSF larvae themselves produce uric acid, which primarily accumulates in the Malpighian tubules. A 5-day exposure to uric acid significantly reduced larval body weight and width, prolonged their developmental period, and substantially decreased both pupation rate and waste reduction efficiency. Furthermore, genes related to oxidative stress were significantly upregulated in the larval gut. How do the larvae cope with this uric acid stress? We hypothesized that gut microbiome might play a role. We compared the growth and developmental phenotypes of germ-free larvae and those reinoculated with a mixed bacterial community under uric acid stress. Under this stress, the growth of germ-free larvae was significantly aided, their body weight plummeted to 24.6% of the control group, and both pupation and waste conversion efficiency were severely impaired. When inoculated with the mixed bacteria, these growth defects were significantly rescued. We further revealed that germ-free larvae were unable to degrade uric acid, whereas the reinoculation mixed bacterial community substantially reduced uric acid levels in the feed. This indicates that the microbiome is essential for efficient uric acid degradation and alleviating its toxicity. 16S rRNA amplicon sequencing analysis revealed a clear separation in gut microbial composition between the uric acid treatment group and the control group. The control group was dominated by Enterobacteriaceae and Wolframimonidaceae, whereas Rhizobiaceae, Bacteroideaceae, and Rhodobacteraceae were predominant in the uric acid treatment group. PICRUST-2-based predictive functional analysis revealed significant downregulation of microbial pathways involved in cofactor and vitamin metabolism, antimicrobial resistance, and protein folding, sorting, and degradation. Subsequently, we isolated several bacterial strains from the BSF larval gut capable of growing in a high uric acid medium. In vitro culture experiments confirmed that these strains possessed uric acid degradation capability. Genomic sequencing analysis identified a key uric acid-inducible gene cluster in the genome of Enterococcus faecalis AHAU24. This cluster contains 5 genes, including YGFK, IgY, and XDHC. Quantitative real-time PCR analysis further confirmed that the expression of these genes was significantly induced under uric acid stress. Based on these results, we speculate that the cluster may be involved in a pathway that degrades uric acid into smaller molecules. Monocolonization experiments showed that germ-free larvae colonized with either Enterococcus faecalis AHAU24 or Escherichia coli K12 exhibited significantly restored body weight, pupation rate, and conversion rate under uric acid stress, accompanied by a substantial decrease in feed uric acid levels. To quickly verify the gene's function, we generated a mutant strain of E. coli K12 lacking the key gene YGFK. The results confirmed that the wild-type E. coli effectively degraded uric acid and rescued larval growth, whereas the YGFK mutant strain partially lost both of these capabilities. This demonstrates that the YGFK plays a central role in bacteria-mediated uric acid degradation and host protection. In conclusion, our results show that uric acid impaired the growth performance of BSF larvae and reduced conversion rate. The gut microbiome mitigated these detrimental effects on the host by degrading uric acid, thereby providing a protective role. Furthermore, we isolated uric acid-degrading bacterial strains from the BSF larval gut and identified a specific gene cluster responsible for uric acid degradation that conferred a protective effect on the host.